Everybody, today we're going to learn about DNA profiling. So what is DNA profiling? DNA profiling is comparing the DNA of one organism with another organism to check how similar the DNA is. Based on the difference, we can find it out whether they are two different species, or they are two different persons, or maybe identical twins, like many examples we can get like this. Now, what are the application of DNA profiling? So one application of DNA profiling we heavily use in a forensic medicines. In forensic medicines, we had to identify the right persons among several individuals. That is the person who is the murderer of that victim. So how we find it out? We need to get their DNA and compare with the DNA of the sample we found, suspected sample of that site or crime scene. And that's one example of DNA profiling, but I'm not going to talk about the detail about how the forensic medicine we're going to use DNA profiling. The aim of the lesson is a DNA profiling, just general theory about DNA profiling. So I just list some application of DNA profiling. Now another one which frequently you are learning in the syllabus is a captive breeding program. So again, captive breeding program is not the aim of the lesson, but briefly, the captive breeding program is a conservation method to protect the endangered species. So we take the animals from the wild into the zoo, and this animals population we're going to increase. So when we're going to increase the animal populations, we need to find it out the cub, the parents, father and mother sometimes, in order to avoid inbreeding depressions. So another example of DNA profiling is the paternity test to identify a real biological father of a child. So next example is a DNA barcoding. Again, DNA barcoding is to find it out the species, the difference of species. So we can use example two different to find it animal species, to find how they're similar or they are different species or same species. For example, we can take a cyclooxygenase COX1 gene, cyclooxygenase C gene, and this gene we can check how they're similar. Or maybe plant, we can take a Rubis coenzyme, which is used in photosynthesis, to check how they're similar. So the idea of this lesson is not to describe detail how we're going to use this in detail in each of the application. Instead, we're going to check the general DNA profiling method, and these are the application. Again, just to summarize, we can use DNA profiling in forensic medicine. So we can use DNA profiling in captive breeding program. So we can use DNA profiling in a paternity test, and we can use DNA profiling in species identification. So now I come to the last and most interesting point. Let's see our closest friend, that's chimpanzees. So when you take chimpanzees, the DNA of chimpanzees and human, it could be 98, 97, roughly. It's very close to 98, 97. It's similar. So difference may be 2 to 3% difference. So we evolved from the chimpanzees like primates. So still we conserve large amount of similarities of the ancestral species. So we can check a DNA profiling of these two and check the difference and similarities of chimpanzees and human. So interestingly, that DNA profiling method can be used to check it. So in the next video, you will learn how the DNA profiling is performed, as well as the background of DNA profiling. So before we going to learn the technique, I prefer what is the background of DNA profiling. So next video, click on to check the background of DNA profiling.